So that's why we think there must be a higher limitation for vegetation growth due to water limitation. So how do we test that? That is the problem, right? Because there is no like a single index. I can say, that index is bigger. That means vegetation is more water constrained. There's no index like that. So how do we do that? So we basically borrow method from this paper, right? So we basically using a simple correlation between vegetation growth data and the water availability data. For vegetation growth, we can use multiple index. We can commonly use NDVI, normalized vegetation index, or we can use some newer product like vegetation optical depth (VOD), or like GPP product from a satellite, or SAFE product. Maybe some of you heard of that. But so it's really just a satellite product to indicate vegetation productivity. And then we can use vegetation water availability data. One we use is SPI. People use as a drought index, but it's actually more commonly you can use that as a vegetation water availability index. Higher number means more water, lower number means less water. So same as the PDSI. So what we did is we make a correlation between these two, right? We look at whether you have a positive correlation or negative correlation. So if we have a positive correlation, we consider this region is a water deficit region. Why we say that is if you have a positive correlation means you add more water, plant grow better. If you have less water, plant grow less. So that's a positive correlation, right? So if you have that, we talk about that means in that region, that's a water deficit region. But if you have a negative correlation, that means if you add water, plant grow less, right? So that means it's not water limited, it's a water surplus region. So that means it's energy limited. So if you think about like a northern region, boreal forest, for example, they are the energy limited system. If you have more rainfall, more water given to them, they won't grow better, they grow worse. So that is a water surplus region. So first thing we do is we classify the global vegetation map, every grid cell. We classify them into water deficit region and also water surplus region. And then we look at the trend over the years, how many grid cells will become from water surplus to water deficit. That's what we are looking at. So, but first we test whether this correlation concept makes sense. We make the correlation and then we look at how this correlation distribution is. So you can see for this map, all the kind of dark color here, they are positive correlation. Right? Most of them are concentrated in the lower latitude. And then when you look at the bluish color, so that's all negative correlation, right? So they are all located in the higher altitude. So, that's already tell us, the correlation actually makes sense. So the lower latitude, that's the kind of dry land, right? So they have a positive correlation. And for the higher latitude, you have a negative correlation, right? So we can also look at this, right? So they are the aridity classification of different regions, arid region and humid region. So the arid region has a positive correlation, humid region has a negative correlation, right? So this is just a first test, say whether this correlation idea makes sense. So it does make sense. Really the more important thing is we are looking at how these correlations are changing. So this is really this map has a two-dimensional information. So you have basically whether they are positive correlation or negative correlation. And then also whether the correlation are increasing or decreasing. There are two-dimensional information. So you can see there are places like a pink color. They are positive correlation, but also the positive correlation becomes stronger. So that means they are water limited condition, and the water limitation become higher, become more severe over the years, over the last 30 years. And but there are also regions, they are water surplus region. and the surplus region become less severe over the years. When we add up every single cell, when we put them together, so that was the general trend over the last 30 years, from 1982 to 2050. So what we saw is the water deficit region, the area become bigger and bigger. And for water surplus region, the area become less and less, right? So that's actually give us the most direct evidence to show, okay, at a global scale, or at least not the hemisphere, so the water surplus region become less, water deficit region become more. That means vegetation growth does become more and more water limited, right? So that is one of the most direct evidence we have. So remember this one we use NDVI, right? So we want to test whether the results is robust. So for Nature type of journal, you have to show your results is robust. You can't just use one index, one product. You have to use multiple products. So then we use different NDVI from different sensor, but also we look at different averaging time and different moving window. But also here I didn't show we use VOD data, we use SAFE data. They all showing very similar pattern really. So here just the same information, but really just telling us regardless what product you use, we're actually showing the same pattern. The water deficit region become bigger, the water surplus region become smaller. And then that's one evidence we have. The other evidence we did is we actually average all the grid cells for their correlation, right? So it doesn't matter negative, positive, we average them across the whole globe. We add up all the grid cells and we looking at the average, how the average has been changing. And what we found here is regardless we use SPI or PDSI. What we found is the correlation has been, the average of the correlation of the whole globe has been increasing, right? So because remember we talk about higher number means, that is more water constraint, right? So that also give us evidence to show that at the global scale, water limitation become more and more severe. So that's another evidence we provided about the increasing water constraint. So last evidence we have is actually the response time, right? What I mean response time, sorry, there's another evidence, but this is the second last evidence. So is the response time. So response time here means, for example, if I have a drought condition, right, the plant won't grow as well, but they could happen in 1 day, but it could happen in 10 days, or could happen in 1 month. Right? So that's what I mean response time. So what we show here is vegetation response time. We look at water deficit region and water surplus region separately. So on the top is the water deficit region. So what we saw is the percentage of a region their response time decrease is much bigger region than the time increase. So what tell us is on average, so the response time in responding to water shortage become shorter and shorter, right? So in the past, maybe 1 month, if you have a drought event, plants start, didn't grow well. Right now, maybe you have a half month, right? So the response time become shorter and shorter. That's for water deficit region. So for water surplus region, it's actually very similar. So the response time also becomes shorter. So remember for surplus region, we talk about if we have a short drought condition happen, it's actually benefiting the plants. It's actually a good thing. But the period of benefiting could be 1 month, could be like 10 days. What we show here is as the time goes by, the benefiting period becomes shorter and shorter. In the past, maybe 1 month is actually benefiting, but right now it's maybe like 1 week, right? So that's also tell us, the water scarcity for vegetation growth become worse based on response time. Their response time becomes shorter and shorter. That's a third evidence we have. Any questions for how we got this? Okay, so the last evidence we did is, so this one, we just picking the, you don't worry about this, so we just talking about, we detrend the vegetation using linear method or non-linear method. They give us similar results, don't worry about that. So, but really the idea here is, instead of using correlation, that's an independent method, we don't use correlation anymore. But we only look at based on SPI, based on PDSI for that grid cell, whether that is a drought year or not, a drought month or not. And if that's a drought month, we'll pick up their NDVI value odd. And if not drought month, we don't worry. And then we do that across the whole globe. And then we looking, so basically what we are looking, if we're looking at only drought condition, how vegetation has been changing. So basically what we saw is if we only look at the drought condition over the last 30 years, so the vegetation index has been declining regardless what you use, SPI or PDSI, right? Regardless is like a linear or non-linear trend. So really the key message here, remember this is totally independent. We don't use correlation. We just picking up the NDVI value when they are under drought condition. And what we show here is over the years, if we under the drought condition, plant growth become less and less well, right? So that's give us the last evidence. So basically we're using 4 pieces of evidence to show over the last 30 years also, as a global scale, vegetation growth become more and more water limited, right? So any questions? Okay. And so far when we talk about water availability, we really talk about rainfall amount, right? We just talk about how much total water we have. So we rarely talk about the rainfall variability, like how rainfall got delivered. But for this newer paper, I'm part of it, we are actually using the satellite information to show actually rainfall amount and rainfall variability both are very important. So this is based on statistical modeling. So, but the idea is basically what we saw here. If you compare rainfall variability impact and the rainfall total amount, so they actually almost comparable in terms of the region they affected. Based on the numbers, 23% is basically like a positive affected, and 19% of area is negative impacted. So it's regardless the number, but really just talking about the magnitude. So the rainfall variability impact is on the same magnitude as the rainfall total amount. So I guess the idea is in the future, when we think about the rainfall impact on vegetation, we shouldn't just focus on the total amount itself. I have to think about the variability. Variability could play a role as well. So I guess this one I won't elaborate too much, but the idea is, if you have a blue dot, that means, they are positive response. Like a red dot, red color basically means negative response. So because depends on whether you are dry land or like you are non-dry land, their response could be positive or negative. But what we are arguing is really the area is actually quite comparable. So not the direction of the response. Any questions for this? So far what we talk about is essentially drought condition, right? How drought impact the vegetation. So the next part I'm going to talk about is just one paper, right? So talk about the flooding impact, right? So this one is the one when I doing the tutorial, remember I mentioned this paper, right? So it's leading by a collaborator from IU, from another campus, IU Bloomington. So for this one, we really talk about the impact of vegetation on flooding events. Right? How that, to me, the key message is flooding will happen more frequently, but vegetation can help us. They can mitigate the impact, make the impact smaller, right? That's the take-home message for this study. So, and I will give you some rationale and also some how do we exactly did that. This is just based on modeling, based on model results, right? It's not based on satellite or any observations. So, I guess we know flooding frequency has been changing just like a drought, right? So because of the precipitation pattern change. And also we know even right now, right? So the flooding events has been negatively impact us in many different ways, right? So, but also most of the time people always assume if you have a higher rainfall events, you must have a higher flooding, right? So that's a kind of common assumption, right? So, but in reality, right? For example, in this paper, so basically what people show here is, if you have a higher rainfall events, this across whole Europe. So when you have a higher rainfall events, doesn't always you have a flooding. Sometimes actually lower flooding, right? Some places sure you have a higher flooding events, some have a lower flooding events, right? So that's basically tell us why that is the case. So that's exactly we try to answer, right? We try to, our hypothesis basically is actually vegetation play a very important role, mitigate the flooding events. Depends on how do they do, is actually will have a flooding or not. So, for this work, we also identify one of the gaps in the past work when people doing the flooding events. So they actually most of the time they do as a seasonal time scale or even annual time scale, right? So, but this is just because if you do long-term trend, that's the only things like maybe available to you. But the thing is, when we think about the flooding, right? So a lot of time that is event-based, right? You have one rainfall events, you may have a flooding events after that rainfall events, right? So ideally, we should actually deal with the flooding as an event base. So, but it's not always available, especially when you think about the global scale, right? So because that will be a very large data set. So that's why when we think about how climate change will impact flooding as an event scale, is still not widely reported. So that's why we did this study try to answer that question. So again, this is a model-based study, right? So that's why this work was originally submitted to Nature Geoscience and it actually was reviewed there. But yeah, I guess the reviewer has a point. That's all the results are from model. How do we know that is the right results? It's a valid results, valid criticism, right? But sometimes, that's sometimes is every study has limitations. So for this particular study, we did use like a model ensemble, but we only use one model. So this is a CESM is basically community land model, right? You may heard of this. It's one of the large model people use, developed in the US and widely used. It's one of the member of CMIP 6, right? So we talked about CMIP 6 in the past. And we use different future scenarios, SSP 3 to 7, and so that has future predictions. We'll get data from every grid cell globally and as an event base. So like we talked about, that's very computational intensive. So we have to like a single computer can't be able to do that. So we basically use so our university has a supercomputer called the Big Red 2. So that is has a very high computing power to be able to do that. So what we did is, we found historical data and we pick up all the 95th percentile of the rainfall events, and also we look at their corresponding runoff. We'll pick up all the data from every single grid cells from a global scale, and also we'll then calculate the runoff ratio. Runoff ratio means the ratio between runoff divided by precipitation. So some places they may have an increased runoff ratio, right? So some places they have a decreased runoff ratio, right? So our hypothesis is really related to the previous soil condition. So if you have a pretty wide previous soil moisture condition, right, very likely if you have a big rainfall events, your runoff ratio will increase. If you have a very dry soil condition, and if you have rainfall events, the runoff ratio may not increase, maybe actually decrease. So basically, we just pick up all the data from soil moisture and also the rainfall data and the runoff data from like every single grid cell, and look for all 30 models, make an average, make an example of all the models, and also look at different scenarios. So that's how do we did this research. So here is a kind of a quick result. So this is a comparison of projected rainfall events, projected rainfall in the future compared to like a reference period. So the key message here is what we saw here is in the future will be much wetter globally when we think about the end of the century. So most of the places is higher rainfall events, right? So that is a projection for rainfall events. And then we can looking at the runoff ratio. So remember we talked about runoff ratio is actually quite surprising. We actually found many places they have a reduction in runoff ratio. Even you have a higher rainfall events in the future, the runoff ratio is actually lower. But there are places has a higher, but many places they have actually lower runoff ratio, right? So why is that? Like I mentioned, our hypothesis is because of vegetation. If you have a higher vegetation cover or greener vegetation, right? So they can take up a lot of soil moisture, make the soil moisture less. That actually can reduce the runoff ratio. So what we did is we did look up the previous surface soil water conditions for every grid cell. So we can say actually this map is very similar to this map. You can, based on the comparison of these two, the soil moisture, previous soil moisture condition always matching the runoff ratio. You can actually look at based on the relationship between soil moisture and runoff ratio. They are scattered, but almost like a linear relationship there. So that's basically tell us soil moisture play a very important role. So we can also use a random forest type of machine learning technique to look at the major contributor for runoff ratio decrease or increase. So you can see the major players for runoff ratio decrease. One is transpiration, canopy evaporation, and ground evaporation. So they are the major players. Right? So the same for runoff increase is also transpiration, evaporation, and ground evaporation. So basically, to put this all together, what we think happen is soil moisture condition. The previous soil moisture condition play a very important role for flooding events. If you have a higher rainfall events, it really depends on whether you have a higher or lower previous soil condition. If you have a lower soil moisture condition, you tend to have probably flooding events. You may not have a flooding events, but if you have a higher previous soil moisture condition, you tend to have a flooding events. So but why that happens? Because of vegetation. So because in the future, when we do the projection, we are talking about in the next 50, 80 years later, right? Vegetation become greener, become bigger. Right? So they will actually mitigate the water loss through evaporation, transpiration, right? And the soil moisture change. So because they moderate the water cycle. So really the take-home message here is vegetation play a very important role in terms of mitigate the changes in flooding in the future. So just a quick summary for what we talk about the flooding part. So what we show here is a runoff generation from extreme rainfall is complex, right? So it actually depends on the rainfall itself, but also depends on the condition, the previous condition of the soil condition. So if you have a drier soil, means you may not have actually flooding events. If a wetter soil, you very likely have a flooding events after extreme rainfall events. So, and also we talk about, even with increase in extreme rainfall in the future, actually majority of the place we saw is actually has a reduction of runoff ratio. Right? It's runoff ratio, not runoff, right? So remember. So, and the mechanism for that we talk about is because of vegetation, right? Because we have a vegetation gradient in the future, and that vegetation gradient will actually decrease the soil water content. So because they decrease soil water content, that actually will prevent flooding events after heavy rainfall events. So really here just implications, right? So if we know vegetation play a very important role for mitigate the flooding risk, that means when we think about the city planning, right? You have to think about make sure you plant plenty of trees, that can help us with flooding condition, right? Even in the future, we know there are extreme rainfall events. If you have a lot of vegetation there, that means very likely the chance of flooding events will be reduced significantly. So to summary both part, right? So for the drought part and the flooding part, really what we saw here is based on the flux tower synthesis work, what we saw here is the drought timing play a very important role for ecosystem carbon sequestration. If you have a drought happen in the late growing season, that's very bad. But if you have a drought happen early growing season, it's not too bad. And then I talk about we develop a multi-sensor based drought index that actually can be effectively capture drought almost as good as USDM, the widely used one in the US. But this one is a drought index, is a continuous value, is remote sensing based. It can be applied to other place, other continent. And based on the correlation between vegetation index and water index, what we show here is in the past 30 years, we think vegetation growth has become more and more water limited. Right? So one of the evidence, another evidence is we saw the response time to drought is much shorter now, and also if we pick up only drought condition and look at what plant growth is during that period, we can see over the years, if you are doing the same type of drought condition, the vegetation growth has become less. And then last, when we talk about vegetation gradient, basically we are talking about, in the future, sure, there likely has more extreme rainfall events, but that doesn't necessarily mean you have to have more flooding, because vegetation become greener and that they can actually mitigate the flooding events. It could be actually have a lower runoff ratio in the future. So, but again, this one is purely based on the model projections, right? So we don't really have a way right now to validate in the real world yet. So I think that's all. I guess I just again acknowledging our funding source from different federal agencies in the US, but also for my group members for their contribution for the work I taught here. So hopefully, I don't talk too fast, and you get the key message, at least more. For the method, some of them is tedious, may not be as easy as you think. But if you have a question, feel free to ask me. But hopefully you get the general picture about the general results and also the implications. Yeah, thank you guys. Any questions, comments? So hopefully that's because everything is clear enough, not because everything is not. It's just yeah. Yeah, that's a good question. I mean, there's no way to validate future projection, right? So the only thing you can do is you can use the, for example, if you find a relationship, you think that will happen in the future, right? So you can only validate the part currently using the data to validate that relationship. If for example, if your model output, for example, for this one, what we did validate. What we validate is, we can't validate the future part, but we can validate like in the current data, we have a, runoff ratio, we have a soil moisture, we can do this relationship for the current setting. So if we can match the current setting and the model results, we have to assume the relationship will stay the same in the future. So I guess, yeah, that's the most commonly people does, is because there's no future observation. We have to rely on, but of course the caveat for that is you does assume the relationship stays the same right now and in the future. But in the future, sometimes the relationship may not hold. That could happen, but that's the best thing we can do. Just using the current observation to validate your model output. And then if the model output captures the current observation, you can say, then I can do the prediction now. Yeah, thanks. Sure. Any other questions, comments? Yeah. So what kind of vegetation? Yes. So, yeah, so the question is, yeah, I guess what kind of vegetation responsible for this? But remember, this is let me see whether I have the spatial scale. So this is a global modeling study, right? So I don't have that, but this is a 0.5 degrees C, like a 50 kilometer by 50 kilometer, right? So in a way, we don't really care or don't know what vegetation is. It's just in general, there are vegetation there. But of course, in reality, most likely we're talking about trees, right? So that's the major player in this. But yeah, for any of the global study, most of the time, the best people can do maybe 0.1 degree. That's still 10 kilometer by 10 kilometer. So that's already very high resolution for global scale study. But even that, you can't really tell too much about the vegetation type. So yeah, for like a regional local study, for modeling study, you can easily do that. But globally, very rare people were able to do that. Yeah. Yes, that's a good question. Any other questions, comments? Okay. Yeah, I guess that's lunchtime now. Then. So yeah, if you have questions, yeah, feel free to come to me separately. If you have a question about certain procedures or just general questions, I'm happy to answer it. All right, thank you guys. you don't know the number is the correct number, right? So, but at least you have to have it. So, yeah, I guess really the point, yeah, just try to think about whatever you can find, right? So, and then validate, yeah, that's what we do. Sometimes, I do, maybe tomorrow, yeah, I'll talk about fall and hill. Nobody measures it, right? So you have to just find the record yourself and try to use whatever you have. Validation definitely is important for otherwise. Yeah. Yeah. It's a common problem, right? So everybody deals with that. Right? So this. So I mean, to me, really, it's a variety of the data. Like, think about soil moisture, think about satellite data for the map and the cultivation, and yeah, I guess whatever you can find, it's really just the variety of the application. The more you find, you generate data that way. So you don't really collect data from the field or doing remote sensing. So that's kind of the the simple way to think about the remote meta-analysis. So why meta-analysis is useful, right? So because it's provide way quantitatively synthesized data, give you the kind of quantitative information, and because you use a very large sample size, right? So a lot of time when you do field research, for agricultural research, for example, you only have a couple of fields, and you have a limited size, sample size, either like a couple years or sometimes even one growing season, right? So for meta-analysis, you try to synthesize studies from all over the place. You could have very different soil texture, different temperature, different crop type. You can actually put them all together. So increase the kind of the power of your conclusion, because it is based on a very large data set, give you a general trend. And also, a lot of time for individual studies, there are different results. And by doing meta-analysis, hopefully you should be able to find a common trend and try to reconcile why these two studies are different. You can give a reason why it's different. So that's why meta-analysis is quite powerful. It's quite useful. So really the key thing, the most time-consuming part, I would say, is kind of digitizing the literature data, right? Because you have to go to every paper, and a lot of time you digitize the data from the figures in the paper. You take them out. There are many software you can use to digitize that. But if you have data already in table format, that's actually much easier, right? You can just read out the data. So here I just give you an example how meta-analysis is more powerful compared to regular, for example, review paper, right? So if you working on review paper on certain topic, maybe whether flooding will, whether whatever, I guess, topic, you have to have a treatment, right? So the treatment could be drought, could be flooding, could be anything, heat wave, right? If you want to say whether, for example, heat wave will change this certain crop yield, you want to test that. So traditional way, when you do a review, you will go to different studies, right? So you will see, okay, this study has a certain data. They get a conclusion, maybe impact crop yield or maybe not. And then you go to a second study, you also get the conclusion, impact or not, right? So, then you go to maybe number of studies and try to say, okay, overall, how many actually are finding the drought or heat wave impact the crop yield for that species, and how many are not. Really, you're just kind of counting the vote, right? Either this is yes or this is no. You just simply count them. So that is, I guess, the most common way, most traditional way you try to synthesize literature data, right? So that is good. That is one way to do it. But the downside is you're really just kind of using the secondary information, right? People already draw a conclusion. You just count how many is the conclusion for 1, 2, 3, and you make your own conclusion. For meta-analysis, you really don't care about the conclusion people made in their own paper. What you do is you really just collect data from each study. And then you combine the data. You don't combine the conclusion, right? So you actually rely, your own conclusion is rely on the combination of all the data, right? So that is a difference between kind of a traditional way to deal with data synthesis review paper compared to meta-analysis, right? So that's make sure you understand the difference, right? So there are many different software can help you digitize the data, but also synthesize the data, analyze the data. So today, we'll basically try to let you have a little bit taste for that process. So, why is very useful? Also, you can actually reflect it in the real outcome, right? So that's my Google profile. That's all the top cited paper I wrote, right? So you can see actually the one in the box. That's one all like a data synthesis paper. So you can see that's more than half, right? Really, I can say majority of the paper highly cited is actually through the data synthesis. So in a way, it's not surprising because you are helping people to synthesize that field. You already help them to make conclusions, so they don't have to read 100 papers themselves. You already gave them the information from that 100 paper, right? So it's actually that's why it's very, very powerful. So that's the paper I talked about earlier today, but there are other data synthesis we did from my group over the recent years. So, but just give you the kind of a practical benefits. Using that maybe will help you write impactful, like highly cited papers. Right? So that's but the difficulty for meta-analysis really is you have to find the right topic. Because if you have a brilliant idea, right, you want to answer a question, and then when you search the literature, there are only 2 papers there, then you can't really do it, right? So, or if you have a topic so broad, when you search for papers, you find maybe 10,000, 100,000, then that's almost impossible to, digest all of them, right? So you have to be able to find a question that is kind of broad enough to be useful to many people, but also not broad enough. You have just too many paper, you can't even handle that. So that part does require your kind of intellectual judgment, right? You have to read things, think about things yourself. What is actually the most pressing question I should ask, and do some preliminary searching, and like just look how many papers in that topic already, and then decide whether it's worthwhile. I can do a meta-analysis, right? So that's something you have to decide. So when we think about the meta-analysis, really there are 2 important things you want to do after you collect data. That is important, it's tedious, but that's not a part of analysis yet. So the analysis one, we basically try to calculate effect size. So what is the effect size? Really just you try to look at the difference between your treatment and your control, So for meta-analysis, because again it's come from the medical field, right? So in a way, you have to have whatever the medicine as a treatment, and then have a control group. So you want to say, when you compare this one, whether they are different or not. If they are different, means this medicine works, right? So that's the essential idea for meta-analysis. So the same idea for other field, really you try to say whether the treatment you are looking whether, for example, if you want to say whether the flooding will increase, whether extreme rainfall will increase flooding in certain climate. You're just looking for like a data from that climate condition, looking for extreme rainfall events, looking for like a runoff, like data, and try to like have with rainfall events, without rainfall events, and looking at the treatment effect. So you have to be able to identify one treatment, one control, and then you make the comparison. But how exactly you make comparison, that is the effect size, right? So one type of way to quantify effect size is called the response ratio, or log R. So really you just take a ratio between these two and then take a log, right? So mathematically is equivalent to difference between log of these two values, right? So x2 basically is kind of the treatment value, right? So whatever the treatment you are looking at, and x1 will be the control, right? So that is your response ratio. So every single study you calculate a response ratio, and then you synthesize all of them. So another way is called HD. So that's another way to calculate effect size. You can see really the major difference between these two is HD does try to incorporate information of the variability, standard deviation. And this one is just a correction for if you have a small sample size, there is a certain way you can correct for that. But really you're thinking about the treatment difference and then standardized by the standard deviation of your study. So this one is less commonly used because many studies don't have the standard deviation reported, and this one is I would say the overwhelmingly more commonly used. But I would say maybe 60% using this, 20-30% using this, right? So when you read the meta-analysis paper, that's kind of the divide. So really, for us, maybe for exercise we try to do today, we will just use this response ratio. So that's one thing. After you digitize the data, after you get your raw data, the first thing you do is try to calculate effect size. Then really the last step for meta-analysis is you calculate the confidence interval, right? The one I talked about earlier, right? So that's one, yes. Okay, so control basically is whatever you consider as a standard condition, and then treatment be some kind of either heat wave. For example, if you want to look at the drought impact on agriculture yield, like the one I talked about earlier, so the control will be no drought, like a normal condition. So the treatment will be drought condition, right? And then if you look at the yield, right, you really just look at the yield under normal condition, under drought condition, and then you take the ratio. So that will be the response ratio. You take R log of that ratio. That will be your effect size. So yeah, really the control and the treatment, it depends on what you try to study, right? So if you want to say, for example, whether warming will increase compound effect. I mean, that's it's hard to do control experiment. You have to rely on control experiment. For example, if I say, if I plant certain trees in this watershed, whether that will reduce the runoff, right? So that if that's your you are thinking about your treatment, so the planting the tree will be your treatment. Then you should find another similar watershed without planting the tree. right? So that's your control. And then if you're looking at the basically runoff value, you can just measure the runoff value in each of them, and then take a ratio. That's your effect size. So yeah, really, whatever the, I guess the impact you want to look at, that's your treatment. You consider that way. Yeah, sure. So yeah, that's good. That's good. Yeah, no. So after you get effect size, really the most important part is try to calculate this thing, the bars, right? So this one, like I mentioned, is a 95% confidence interval, right? So typically, that's statistically that is the range of your treatment effect, right? So if this one is is overlap with 0, that means there's no change. The treatment and the control are the same, right? If this is like this, there's no change. So that means your control and your treatment is the same. But if you think about the drought impact on yield, means okay, drought didn't really change yield, right? So for this particular case, that's a paper we are going to use data from. That's we actually looking at the cover crop impact on agriculture yield. Right? So whether if you use cover crop, whether that's increase or decrease or doesn't change your yield, that's our question. So here, basically, if you have things like that, that means cover crop really didn't change yield. And if you have things like this, that means cover crop increase yield. And if you have things like this, that means cover crop decrease yields because they are on the negative side, right? So that's basically the end product you try to get. So before I move on, any other questions? So if I try to understand the impact of a cover crop on yield, what is the treatment, what is the control right now? So I will have one field with cover crop, one field without cover crop. Which is control? So always the one without, the one without cover crop should be control, right? So that will be our reference value. And then the one with cover crop should be our treatment, because here cover crop is your treatment, right? So same thing if I try to look at, for example, simple question, right? If I want to say certain fertilizer works for the crop yield, right? So I have two fields, one will be adding the fertilizer, one will be not adding the fertilizer. Which one is the treatment? Crop yield, yes. So I guess in terms of one adding fertilizer, one don't add fertilizer, which one is the treatment, which one is the control? So the one adding fertilizer, that's your treatment, right? So the one without the fertilizer, that's your control, because you want to compare these two, right? So just make sure you understand that part. Any questions before we move on? So, for example, if I try to study, for example, how rainfall variability will change crop yield, if that is my question, right? So if I look, then I need to look for studies that actually change rainfall pattern, right? And if I have two field or two plots, one actually people change rainfall arrangement, one is not, it's just a normal rainfall. Which one is the treatment? The one changed, right? So the one normal condition, that's always your control. And then you take the ratio between these two to get your response ratio. And then after you get your response ratio, you actually calculate this. How do we get this, right? So because this one is not from a single study, this one depends on whatever you study. Could be from 10 study, 20 study, 100 study, maybe even 1,000 study, right? So that is actually a synthesis part. So, but each one, for each individual study, they only give you one response ratio. So what you do here, confidence interval, you really just synthesize all of them, getting the final product. This is the final product, right? So then we can, think about have a try, right? So there are different software can help you doing meta-analysis, right? There are a language, like a package you can do that. But I think for this tutorial, we'll use one easy to use. That is like Excel. Is you can search MatWin and look for this website and try to look for this software. There is a download button here. So they have both PC version and Mac version, right? You can download and then install on your computer, and then we'll start from here. So yeah, try to find this software first, and then we'll give you a data set, and then you can play with the data set and try to see how can you make things happen. So yeah, first thing, try to look for this software and try to install this to your computer. It's a free software. It's open access software. Anybody can download, can use. Yeah, if you don't have a computer, try to pair up with someone who has it. At least you have a experience, just running the software. So yeah, that's a web link. Or if you Google MatWin, you should be able to find it too. So get it? Good. Installed? Okay, good. Everybody is good so far? Okay. So this one is one of the commonly used one. Yeah, if you start with meta-analysis, you can start from this this one. You can use this one for final publication too. So for interval data set, because it's good to have a standard you can compare your results with, right? So let's just use published papers. So that's the most recent ones from my group. Like I mentioned, that's one we actually looking at the cover crop impact on crop yield, right? So this one published last year. So try to Google this one, and I'm asking to do this one because we did publish the data too. When you go to the website, you should be able to find the supplementary material. That is whether you did it correctly or not, right? So yeah, just try to Google this one, and you should be able to find the data file. There will be a supplementary file associated with this paper. You can download that Excel file, and we'll use that data as our exercise. Yeah, so a good example like this paper published last year, right? I think it's already been cited for almost 30 times. So, it's really just tell you if you're actually doing some like a good synthesis is really good for other people to use your work. Yeah, you can use Google Scholar to search this, and there should be an Excel data file associated with this publication. Do you guys have a issue to access the data set? You found it? No? Found it? Download the data already? Okay. So yeah, if you find the paper, you need to scroll down all the way to the bottom. I think will be a supplementary table, right? So that is the one we provide all the data. So we try to, when we publish paper, we try to make them open access. Everybody can check the results, can use the data. So there is a data set basically associated with this paper. That one already digitized everything, right? Everything is already done for you. It's already there. We basically just play with that data, and that data table should be, part of the file. If you go, so you didn't find the Excel. Sorry. Yeah, let me go closer. So you said there is. Oh. Okay.
There is one problem which is related, seems like that it is related to this problem. That is something we call it as bibliometric data.
Okay.
So in bibliometric data, what we do is we take all the publications.
Sure.
From on a specific research paper.
Yeah.
And then we try to find out what were the important keywords. And what is the important findings.
Yeah.
How the themes of that particular field evolves over time.
Yeah, the evolution. I know what you're talking about.
Yes.
That is based on the text, right? So you actually digitize all the text and you make,
We
yeah.
process the text and then. So is that problem has to do something with this one, or we are only extracting the data and we are performing the qualitative analysis, quantitative analysis?
So for that one, I think, it's less used really, because that one is based on, analysis frequency of the data. Depends on algorithm, right? They may do things good, maybe not, right? So there's some subjectivity there. So this one is really just building on the actual data. You synthesize actual data, so it's more objective. So that's why this one is tend to be, I guess, more commonly used.
Different kind of results than that thing.
Yeah, I think so. So because that one is, in a way, it's not quantitative information, it's more qualitative information. So it's like which field is a hot topic, and who did what, was evolution, right?
One kind of that analysis. That's why I thought, because I haven't heard about this one.
Okay. Yeah, I mean they are related, but totally different techniques. Yeah. But I know what you're talking about. Yeah. So I just try to check how. So you guys find the data? Yeah. I think the university should have you should be able to download the data. I just wait a few minutes before I download the data. Any questions for the data? Everybody downloads the data? Okay. So yeah, I guess I can show you. Let's see whether I can. So if you open up the software, it's something like this, right? So I guess one of the downside for this software is not data input friendly, right? You can't even copy paste from other place to this. You have to load the data, right? So that's why you have to rely on the Excel you have, but you need to make some modification. First of all, this one, when you load the data, it doesn't load the Excel data. It only load like a CSV file, right? You have to convert. So for example, that will be the data set you guys got, right? When you open up something like this, right? So there is like a more than 1,000 data record. Yes. How do you prepare the data? So this is a digitizing, right? So that one, let's say, so basically each data entry you have to entry yourself, right? So that is actually tedious part. That will take several months to build up this data set, right? So you have a article title, you can just record as many information as you want. Depends on, maybe some information useful, some not. But really, most important information is the sample size of the study, and also in this one, CC is a cover crop. So this is the treatment, and this NCC is a control, and all the numbers here are the yield. So this is yield information. So each show that is one study, right? So that's some maybe from same study has several entry, means they could have several treatment. Like for example, some add fertilizer, some didn't add fertilizer. For that, we'll treat them as a separate entry. So, but in general, if they only have a cover crop treatment and the non-cover crop treatment, one study should have only one row. Manually. So that's why meta-analysis, as powerful as it is, it takes time. It still takes time. For this paper, we spent probably 2 years. This so that is from the paper, right? The latitude and the longitude. Is that your question? So yeah, all the information we get from here is from the paper itself. If they report longitude, latitude, we put it here. If they don't, it's empty, right? So because it's not like essential information, we have to need them. We really the only 3 columns we need is the sample size, the yield for cover crop, and yield for non-cover crop. All other information would be supplementary information. In a way, for example, whether they use herbicide or not, whether the soil texture is clay or whatever, and is a dry condition or is a wet condition, what's their annual rainfall. So this is important only later when we try to group them into different groups. I will look at maybe dryland altogether. I will see how many I know is the dryland information. I make a subset of them and analyze them. And maybe I will look at how fertilizer impact this. I will look how much fertilizer they have in each column. But not every study has them. So a lot of other things will be subset of the whole study. So for this exercise purpose, let's basically try to copy out these 3 columns. Sample size, this one and this one, and copy them out. Maybe put into a new Excel data sheet. And the reason, yes, because somehow if you read directly from this file, there are error message. So let's just copy out them to a new file, like this. I think that's what I did in the morning. So so I just copy out the 3 columns, one sample size, and then the treatment. I add one column called 1, because in MetaWin software, you have to have a standard deviation to be able to run the software. But because most of study don't report that, we just give them the same number, really just the same weight. It doesn't change the results. You just kind of, but then you can run the software. If you don't have that, you can't run the software. So just copy the 3, this 3 column. I just copy out this 3 column and then add a column all like 1, right? Make sure, yeah, you do that. And then after that, I basically have a test file. And then I have to save this as a CSV file. Otherwise, MetaWin won't read it, right? So make sure you try this. And then after this, I can input the data using the text file, right? I can load the data. My screen actually is dead, so but I can see it here. So this one I think is separated by commas. So then you can. So that one you load the data into MetaWin, right? Then you can calculate effect size and also calculate the bar, the confidence interval. So I will wait until you get this part. So remember, right now we have a 4 column. This column is the sample size, right? So basically, that is the information you get from the paper, individual paper. So these 2 are the crop yield. So this is the control. This is the treatment. Or maybe I forgot which is which. Let's say. Yeah, this one is the treatment. This one is control. So column 2, this one is treatment. This one is the control. And then this one, we just put the same number. Doesn't matter 1 or 2 or 3. We just put the same number for all of them. So everybody get here? Okay. So after that, you can then calculate the effect size and the confidence interval. Right? So this one, you can say compute. There is one you can calculate effect size. So this one you have to identify what effect size you try to calculate. Right? For us, is the response ratio. You select response ratio here, not HD. Remember HD, you have to have a standard deviation, which we don't. We just use 1. Right? So then you have to specify where is what's the mean value, standard deviation, and the sample size, and what is the control, what is the treatment, right? Remember, we talk about our control is column 3, our treatment is column 2, right? And then the standard deviation is the last one, 4. And sample size is column 1. So yeah, make sure you check response ratio and then make them correctly. So when you hit OK, you will see there will be output tab right now show up. after, okay. So, did you guys get this? Okay. So after you load the data, what I did is, oh, not this one. You can go to compute. So there is effect size. Or you can use this button. Should be the same. You have to save as a CSV file from Excel and then load the data. Yeah, there is a load button here. Only CSV file. Yeah, if you use an Excel file, it doesn't give you error message. Here. Okay. We'll continue. Okay. And so yeah, here like I mentioned, you have to make right choice, right? Response ratio, and then identify them correctly, right? So I already did. I don't do it again. But this is 3, I think. This is 2, and then this is 4. This 1, and then you hit OK. Because I already did, I don't do it again. It should be the output one. You should be able to get this one output. Everybody get this? So for the output, you can say it give you the effect size here until 1,000 something. So basically, each data record, it will calculate effect size for you. So far, so good. So after this, that's the effect size, right? So that's not the final product. So the final product would be the analysis. So there are more complex version, but let's just use the basic meta-analysis. So that one, you identify what your effect size is, right? For us, it's response ratio. You just do response ratio, and it's log transform. So you don't need to change anything, and then OK. And then here, you have to click bootstrap. Bootstrap, statistically, if you have one value, right, they bootstrapping like many times. You can, default is basically 1,000 times, but you can do 10,000 times, like make the number more accurate. And yeah, that's the only thing you do. And you can hit OK. It should be calculate the bootstrap CI, or yeah, there are some corrected ones. Should be very similar. So that will be our final number, this one. So that will be the confidence interval we are talking about. So that will be the response ratio. That's if you convert to regular, it will be like this. So that will be the one you are using to plot. So that will be this one. So you can use any other software. So this one doesn't, the MatWin doesn't plot very well. You can use other software. But the number gave you this mean value, and this is 95% like a confidence interval, right? So let's see. So that's basically from 2% to 4%, right? So then that's, for each individual record. Sorry. So that is overall results. So should be very similar to the ones reported in the paper. But remember, this is based on bootstrap calculation. That means each time you run this, it won't be exactly the same because it's just a random calculation. So, but the magnitude should be very similar. Maybe a small change in numbers, but should be very similar. So for this particular one, this number should be very similar to this one. You can say that's 2%, that's 4% ish. Right? So that's the range you should look at. So basically what we did so far, we just generate this guy. And if you want to generate others, you can use the same data set. For example, that's the overall results. If you look at legume only, you can go to that data set, just look at legume, and then run that again. You should be able to get this. And if you use like the others, you can try. So I guess maybe I will let you guys yourself try to maybe run legume on your own. Get the data set, load into the software, and try to run it. Hopefully you'll get these results. Then you should be good. So again, that's the difficult part is collecting the data. But after that, you should be able to run this, generate figure like this. So that's the procedure. Any questions? So can you speak louder? Sorry. Okay, sure. So just repeat the procedure again. So you mean like run the software or just talk about the procedure? Okay, software then, right? So again, the first thing is after you download the data, right? So I copy out these 3 columns and I just create a new file, just being simpler. So that's the new file I created. That's a 3 column, and then I add and then I save this as a CSV file, right? Because otherwise you won't do. And after I do that, so this one I have to, let's see whether I can clear all the data. Because the problem is it will be, maybe I will try just load this way. I will just load again and see what happens. So I use commas, so that will be separated better. And then, okay. So so that right now I read in all the data. So after this, you can go to effect size, this one, or under compute there is an effect size, right? So you do that, and then really the important thing is make sure you click response ratio, not HD, and then you identify the column. The mean value is this is the mean value, right? So that's all the column are mean value. So control is 3, treatment is 2, and the standard deviation, that's the one we create like the 1, right? So that's the standard deviation, and then sample size is 1. So just make sure you identify them correctly, and then you hit OK. Should automatically give you this is your data. So you create a new tab. So that one has all the response ratio here. And then after you have this, only after you have this, you can do the meta-analysis, the analysis tab. So you click analysis, right? So we run basic meta-analysis. And then really the only thing we use response ratio. The only thing you need to identify, click is a bootstrap, right? So you can make this bigger number, like it take longer, but the results should be more accurate. So after this, it will give you, so this will be the essential information you want to have, right? You can say the range is actually very similar if you use 1,000 tab or 10,000 tab, right? So this will be the bar, and the mean value, right? You have that's the mean value 0.03, and then the confidence interval is from 0.2 to 0.4, right? So you get that bar, right? So that will be really all the procedure you've done to be able to get that. And again, if you want to try again, you can look for like only legume, right? Like mentioned, in this data set, we have all the information here, whether they are legume or not, right? You can you can see there is a cover crop family, right? You can actually look, maybe just select only legume, right? So you should be able to, so all this data are legume, and then, but you can basically still doing the same thing, but only select the legume part, right? So then you run the same analysis, you should be able to get a different bar, right? So that's how do you do it? So again, the data set is already there. We already built that. But if you do your own study, really the time consuming part is really how do you build this data set, right? So that will take time. So, I mean, for this particular case, we know which column represent for, right? So we know what they represented. So if you have your own data set, you just need to label them, remember it. So, because when I do, this is 1, 2, 3, 4, really, in the software. This is 1, 2, 3, 4. So this is column 1, this is column 2, this is column 3. Yeah.
So that is what I am getting. Column 1, column 2, column 3.
Yes.
After that, since situation is also column is 4. Why?
Why there is a 4?
Sir, he is asking about the software that column 1 to the exercise calculation. It's exercise.
Yeah, no, I know what you mean. Yeah, you are talking about here, right? Hmm? Right. So this is based on. Yeah, so that's based on your data, right? So because here I know, again, I copy this one. This is column 1, column 2, column 3. Remember, right? So that's what they stand for. This is my column 1, column 2, column 3. Column 1 is the sample size. That's why I select when I do sample size. You see? Sample size is column 1. Standard deviation is column 4. Because this one is the one I added. And this one. So this is a 3 because the 3 is the treatment. The 3 is a cover crop, remember? This is a column 3. That's a column 3. Because this is a, oh, sorry, no. Actually, no, this is a, no, this should be control. Column 3 should be control. So this column 2 should be treatment, right? Yeah. So I think we did it correctly when we ran it, but here, yeah. So this one, 3. This is 2, right? Yeah. So yeah, we ran it correctly, but here. So that makes sense now. So it really doesn't matter which one. It's really based on what you actually put the data. You know which one is the treatment is your column, which one is control is your column. That's just up to you. But for this particular case, because we know which is treatment, which is control. Any other questions? Does this make sense to all of you guys? Right now you know what it is? remember the column 4 is the one I just created in another file, right? So that's a column 4. It's just yeah, just same number. You put any number there. Because when you digitize the data from the individual paper, very few will report you the standard deviation. You don't have that information. That's why you give them the same number, same weight. It doesn't change anything. But you have to have that, because if you don't have here, if you don't specify standard deviation, if you don't have a number, it won't run. You have to put a number there. But in our case, we just put the same number. Any other? Yeah. Yeah, so really what we do is, for this one, right? So I guess, you can say, for example, if there is a study, like, when you read the method section in the paper, it will tell you, for example, exactly what the keyword we use in Web of Science to actually search all the papers, and then what the criteria we use to filter the papers, and then whatever left over, that's the one we are going to use. So, maybe 100 paper, maybe 200, maybe 50, right? So we have a list of a paper we will use. So, and then after we have that list of a paper, for each individual paper, I will put it out and then just record the information from that paper, whatever they have, like where the location, where, what the cover crop species, what the fertilizer they use. Some maybe have this information, some don't. If they don't have it, we just keep it open, keep it blank. You just keep doing for all of them. So again, the only important part is really these guys. What's the sample size they did, and then what's the yield they produce. That's the most important information. And then you just keep recording every paper. And then after you maybe go through 100 paper, you build this data set. So does that answer your question? You're clear about the procedure? So the tricky part is, not every paper report everything, right? There will be a lot of open space here, blank space. Some, you can say it's not evenly distributed, right? So some people have this information, cover crop nitrogen rate. Some doesn't, right? Doesn't matter, because later when we do different groups, we only select things. When they report this, we'll put them together. We'll run meta-analysis, and we'll have a confidence interval for that particular thing. If they don't, we just can't report them. So any other questions? Yes. Mm-hmm.
What do you mean by sample size? Sample size. What is it?
So sample size is, for this particular case, that will be, so that's the one they report in the paper. They will say, I have 4 replicates, and that sample size is 4. So they, if they do cover crop impact on yield, they won't just do one field. They will do like maybe 4 plots. So they will say their n equal to 4, and we'll just put n equal to 4. So they will tell us in the paper. We just read that number and put here. So how many replicates essentially? How many replicates they use? Yeah, some people use 3, sometimes use 4. That's a very common, either 3 or 4, right? Because only 3 you have a standard deviation, right? Any other questions? with areas. Oh, you mean like where? How do you apply this method to what kind of area? So, I mean, essentially it could be any area, but you have to be able to identify control and treatment. You know, essentially they have to have some kind of treatment. Yeah, you can do forest, right? You know, like a lot of people, you know, if you increase CO2 for that forest, right, you want to see how that change hydrology, right? So your treatment will be CO2 enrichment, and then you have a control. And the forest hydrology will be either, you know, stream flow, or could be like soil moisture. You know, you can do any of that. So, and you can do anything. Yeah, it's very broad. So I would say, like, if you're familiar with the procedure already, if you are comfortable, I will encourage you to do that on your own for the legume one. Like if you know, you can compare that with the results from the paper. You know whether you did correctly or not. But if you don't want to do it, that's fine. So yeah, I guess that's pretty much for the tutorial. If you are, yeah. So, I mean, ECC just means like a cover crop treatment here. And NCC is a no cover crop treatment. Because, yeah, I mean, that's our own labeling. It doesn't make sense, I know. I mean, yeah, for me, yeah, somehow my student use this one. You know, you can use whatever makes sense to you, right? I would say I would use T or C, probably make more sense, but because a lot of time people use E because enrichment, you know, like a lot of treatment is enrichment. That's why meta-analysis, a lot of people, they use E and C. But there is no requirement, whatever you use, as long as it makes sense to you. So yeah, I mean, so right now it's basically up to you, right? So if you are comfortable with the